Welcome to my another session. In this session, I have a talk about elongation of transcription in prokaryotes. At the beginning, we came to know that the RNA polymerase which binds at the specific site which is known as the promoter site, and such promoter site should have the start point, that is the +1, and the -10 and the -35 element. Suppose that this is the RNA polymerase enzyme. The sigma factor of RNA polymerase enzyme will interact with the -10 element, and which causes the unwind of DNA strand or the melting of DNA strand, and which forms the transcription bubble. So this is the transcription bubble, and this is the RNA polymerase enzyme. With the formation of transcription bubble, the initiation of transcription starts. So initiation of transcription with the formation of phosphodiester bond in between first two ribonucleotide triphosphate and releasing of pyrophosphate. So this is the overview of transcription process. At first, the RNA polymerase enzyme binds with the closed binary complex. After the binding, with the unwinding of the DNA strand, which forms the open binary complex. With the initiation of transcript, or with the initiation of RNA molecules, it forms the ternary complex. So this is the idea. And RNA polymerase, which moves in forward direction, means towards the downstream, with the continuously lengthening of RNA or synthesis of RNA strand. Suppose that this is the DNA strand and this is the transcription bubble. And such transcription bubble which moves, or which have to move from the start point to the terminator sequence or to the terminator. So with the movement of transcription bubble, which forms supercoiling at the downstream region, and negative supercoiling forms at the upstream region. So this is the, suppose that, upstream region. Such supercoiling or positive supercoiling, actually it relaxed by the action of one enzyme, that is the gyrase. So the action of gyrase enzyme, which relieves the positive supercoiling and aids the negative supercoiling. Likewise, by the action of another enzyme, that is topoisomerase, which relieves the negative supercoiling and relax or restore the original tone or original coiling of the DNA strand. So this is the process. At any time, with the continuation movement of the transcription bubble at any time. Suppose that this is the RNA polymerase enzyme. At any time it is seen that suppose that this is the DNA strand, this is the template strand, this is the downstream DNA strand, this is the upstream DNA strand. The downstream DNA strand enter into the RNA polymerase through the downstream DNA channel and exit through the exit channel. And suppose that this is the newly synthesizing RNA molecule which exit through the RNA exit channel. So here the synthesis takes place from the 5' to 3' direction. At any time, such RNA, such nascent RNA bonded with the template DNA by 7 to 9 number of base pairs, and the remainder nucleotide which continuously peeled out from the template strand. So this is the idea, and the newly incoming nucleotide come and aids at the 3' end of the newly synthesizing transcript by the phosphodiester bond with the removal of or releasing of pyrophosphate. So now, how the translocation takes place? So to describe the process of translocation, there are two models which propose the translocation of RNA polymerase during the elongation process. So what are these? First is the monotonic process of translocation, and another may be the inchworm model of elongation. So in case of monotonic translocation, according to this process, the RNA polymerase which moves forward by one base pair. So single base pair or single nucleotide is available at the active site for the RNA polymerase to copying or to transcribe, and this is known as the monotonic process in which the transcription takes place continuously. But according to the inchworm model, the RNA polymerase enzyme which moves by 10 nucleotides. According to this inchworm model, the RNA polymerase it has two regions. It has two regions. The front portion, the front region, which is known as the leading product site. This is the known as the leading product site, and the end portion at the back portion of the back region, which is known as the lysing product site. According to this model, the front portion which first move 10 nucleotide of DNA template, but the lagging product site which tends to copying the preceding nucleotide. After the completion of copying the preceding nucleotide, such portion compressed and the front portion or the leading portion which release and restore the footprint of the total length of RNA. So in simply we say that at a time the 10 nucleotide base pair copying and releasing. So again 10 base pair or 10 nucleotide which actually enter into the RNA polymerase site, and after the copying 10 nucleotide release the 10 nucleotide, and again another 10 nucleotide enter into the RNA polymerase, or the once only the 10 nucleotide copied and release the 10 nucleotide. So here the copying of the nucleotide discontinuously or the formation of transcript takes place discontinuously. The inchworm model, it is not accepted by all. Such model actually it is proposed by M.J. Chamberlain in the year 1993. So monatomic process, it is highly accepted. According to this model, the synthesis of RNA takes place by one nucleotide. It means one single nucleotide add to the 3' of the transcript. So how actually, is there any guide to available of one nucleotide once? Yes, there is some guide, there is some bridge, there is some guard which actually guard or which actually provide the single nucleotide to copy for the RNA polymerase enzyme. So the crystal structure of RNA polymerase enzyme which reveals that the portion of RNA polymerase enzyme which acts as the bridge-like structure, Suppose that this is the template strand and this is the newly synthesizing RNA transcript from prime to 3' direction. At a time, there is the single nucleotide is available for the RNA polymerase to transcribe. This is the protein bridge. Such protein bridge present at the uptake nucleotide channel of the RNA polymerase enzyme. These are the nucleotide of the DNA template strand. Those are present at the outer side or they clipped outside. It has two conformational structure of the protein bridge. At the beginning of the translocation, the protein bridge has the straight structure. Now, the single nucleotide is available for the enzyme to be transcribed, and it is transcribed. Suppose that it is transcribed here. Then now the protein bridge have to translocate. So now the protein base try to translocate. So at the time of movement of protein base, the protein base become band-like structure. So this is the bending structure of protein bridge. So with the bending structure of protein bridge, allow the another nucleotide to enter at the active site. That's why those nucleotide cleave out. Now such nucleotide clipped inside, which will be copied, and now after the entry of such nucleotide, after the cleave in of the nucleotide, the protein bridge restore the straight structure. So this is the one model. So this model tells that the RNA polymerase guide the entry of nucleotide, ribonucleotide triphosphate, by the formation of protein bridge. Such protein bridge which move or which allow the another ribonucleotide triphosphate by the bending structure, and again it restores the straight structure which abstract the entry of another or further ribonucleotide triphosphate. This is the model. So with the movement, with the translocation of RNA polymerase enzyme, such enzyme, it works differently. For example, the RNA polymerase enzyme which continuously forms the base pair, continuously which forms the phosphodiester bond, and another additional function also takes place by the RNA polymerase enzyme, that is the proofreading, the searching of incorrect nucleotide or searching the mispair of nucleotide, and which is known as the proofreading process. So the RNA polymerase which search the incorrect bases, if there is some mispair takes place, then RNA polymerase caught and remove out from the transcript. This is known as the proofreading. Such proofreading actually takes place by the two processes. One may be the hydrolytic process, and another may be the pyrophospholytic process. In hydrolytic process, the editing process takes place. Suppose that this is the newly synthesizing RNA. If any base pair found too incorrect, then the RNA polymerase enzyme which moves backward and cut the DNA strand and remove the portion of transcript from the template strand. This is known as the hydrolytic process of editing. So in this process, the RNA polymerase enzyme which moves backward by 2 or 3 or more nucleotide, then cleave the RNA and release the RNA from the template strand. Suppose that this is the template DNA strand. And the some model or some workers, they proposed that there is the involvement of grief factor at the time of hydrolytic editing. So another process may be the pyrophospholytic. As you know, the phosphodiester bond actually takes place by the attack of 3' hydroxyl group to the incoming nucleotide alpha phosphate bond at the 5' region and the removal of pyrophosphate. So if you add the pyrophosphate again, then there should be the breakage of the phosphodiester bond. So in this idea, such pyrophospholytic editing takes place. In this process, the addition of pyrophosphate takes place. By the incorporation of pyrophosphate, the phosphodiester bond will break, and by this process it may remove the correct or incorrect base pair. So the RNA polymerase, it continuously performs different functions like the base pairing, the formation of phosphodiester bond. With this, the additional function is the proofreading process. So how actually at the time of translocation, at the time of elongation, how actually these strands contact with the RNA polymerase? There are one model which proposed that at the time of translocation, at the time of elongation, sufficient contact takes place in between the RNA polymerase with the substrate nucleic acid, that is the template DNA strand, and the product nucleic acid, that is the RNA transcript. Suppose that this is the RNA polymerase enzyme, this is the DNA template strand. Such DNA template strand transcribing from the 5' to 3' direction. Suppose that this is the RNA. Some portion of the RNA polymerase enzyme which contains some specific amino acid which interact with the template strand. It means that the RNA polymerase, some portion of the RNA polymerase which forms the bond with the template strand, and some portions also form the bond with the RNA transcript. So at the time of translocation, these bonds are broken. So this is the template strand, DNA strand, and this is the RNA transcript. So at the time of translocation, these bonds are broken. So the interaction from the RNA polymerase will break. So this is the breaking interaction from the RNA polymerase with the RNA as well as the DNA template strand. So after translocation, again the interaction or the bonds are remade. So suppose that, so after the translocation, again the bonds are formed. Suppose that this is the template strand and the translocation takes place. After the translocation, again the bonds are formed. So in this way, breaking and remaking of bonds takes place with the RNA polymerase, which the substrate means template strand and the DNA strand, which stabilizes the transcript and the template strand with the RNA polymerase enzyme. So this is the total idea about the elongation process. So before going through this elongation process, or prior to entry into the elongation process, the sigma factor from the RNA polymerase will dissociate. So first, the sigma factor dissociate from the RNA polymerase enzyme, then the RNA polymerase enzyme enter into the elongation reaction process. So if you like, then click on my another video to get the termination of transcription in prokaryotes. Thank you.